This is a production of Cornell University.
So I'm Gennaro Fazio. I'm the apple rootstock breeder for the joint breeding program between the USDA-ARS and Cornell University. The program was established in 1968. That's close to the year when I was born. So I'm the second generation breeder in this program. And apple rootstocks. I bet only a few of you know what apple rootstocks are. And so I'm going to demonstrate a little bit the concept of apple rootstocks and their importance. First of all, this is an apple tree. It has apples hanging. This is actually a Fuji apple tree. But as you can see, you go all the way down to the bottom right here, and you look at the bark, and the bark suddenly changes to a different type of bark. This is the graft union. This is the rootstock. This is called a scion. Of course, it's joined by grafting. So we're actually breeding the root system and this first part of, we call it the rootstock shank or trunk. And what do we breed for? Initially, we breed for disease resistance. Then we look at other parameters like yield and dwarfing, early bearing, also called precocity. We look at mineral nutrient absorption by the root system and their translocation up into the scion. We have ventured into also selecting for improved winter hardiness, and also we can select for improved nutrient balance. The breeding program was initially established because of a scourge that's North American in origin. It's called fire blight, and if I go right over here, I have a little example of what fire blight looks like. Right here, you see this little shepherd's crook. This is the typical sign of an apple tree that has been hit by fire blight. The problem with fire blight is that, okay, fine, it hits up here in the top part of the tree in the scion, but fire blight actually travels all the way down to the rootstock in like a couple weeks, maybe sometimes 3 weeks. And if the rootstock is sensitive, it just dies. So I've seen orchards with thousands of trees that were grafted on one of our traditional rootstocks that has been around for about 300 years, called M9, and I've seen thousands of trees dead because of this disease, fire blight. So that's one of the reasons why the program was established. It was also established to do something else, and that is be more dwarfing and more productive. So one of the big traits that we breed for is dwarfing. This tree right here and this tree back here were planted at the same time. The only difference between these two trees, it's the same scion, it's the same scion variety, but the only difference between these two trees is the rootstock and the rootstock genetic potential to dwarf the tree. We've discovered the genetic loci that contribute to this dwarfing character, and we currently use marker-assisted breeding to select for those in our populations. But when I was talking about dwarfing, the major dwarfing rootstock M9 is highly susceptible to several diseases, including fire blight and replant disease complex. And so the breeding program was established, and it has perhaps the only apple rootstocks that were bred for resistance to fire blight and replant disease complex. So in addition to dwarfing, we also select for a related trait, which is called early bearing and yield or crop load. So as you can see with this young dwarf tree, we can count 1, 2, 3, 4, 5, 6, 7, 8, you know, maybe we have, we're hanging about 30 to 50 apples on this tree. Whereas this more vigorous tree on a different rootstocks does not have the same density of apples and flowers. And that is another trait that we can select for in the root system, in the rootstock, but then it exemplifies itself or displays itself in the top part, which is the scion. So you can imagine now the complexity of breeding apple rootstocks. We're measuring most of the traits on a different genotype. If you can imagine joining two genotypes together, two apple genotypes, you have one that has certain characteristics in terms of hormones and gene expression, also the way the tissue is arranged, and then it's joined with another genotype that has completely or somewhat different characteristics in terms of how it perceives those hormones or perceives water status or something else. And so whenever you make one combination and you measure everything like yield and precocity or early bearing in one combination, things may be slightly different when you join in with another scion variety. So something that is good for Fuji apples may not be so good for another type of apple like Gala or Honeycrisp or you know, some of the new ones, Snapdragon from Cornell. And therefore we have to somewhat account for these differences. It becomes pretty complex. 20 years ago, up to 20 years ago, we used to have a monoculture, pretty much a monoculture of M9 rootstocks. That was the rootstock that was applied in about 90% of newer dwarfing apple orchards in the US. We've been able to transition through by releasing new Geneva rootstocks that are disease resistant and more productive. We've been able to transition to a much more diverse portfolio of apple rootstocks. So we have about 12 apple rootstocks that are program released, and they all have fairly different applications. Some are more vigorous, but yet still very yield efficient, and some are less vigorous or promote a smaller tree, but are able to produce larger fruit in certain cases. We have come up with a concept that's called designer rootstock, or a rootstock that's designed to fit in certain circumstances. For example, we could use a very different rootstocks here in New York compared to Washington State, and then compared to Brazil, where we actually do have a good production of our rootstocks, and then compared to South Africa and California and Pennsylvania and Maryland. Why? Well, because soil quality is different, or the soils are different. The water quality and the amount of water available is also different. This orchard right here is not irrigated, and it's completely reliant on natural rainwater. Whereas most of the orchards out west in Washington State, Idaho, and Oregon, all the way down to California, require irrigation. And so that is also a something that we have to account for when we breed, because it's the root system that absorbs the water, the nutrients and everything else and sends them up to the scion. And so how that irrigation is done is influencing the way a rootstock performs. So with apple rootstocks, we're actually breeding two different crops. And why do I say that? Well, we're breeding to generate these little things, these little sticks. These are actually rootstocks. And so we have a point in the nursery where we propagate these. We can propagate by micropropagation, but also by stooling. And so the stooling properties are also important. So we breed for that part of the growth cycle, where we have mother plants that are cut down every year to produce these liners, rootstock liners. And then we breed for the phase in the orchard. So they move from a rootstock nursery to a finished tree nursery, these rootstocks. They get joined by grafting and budding, and I'll demonstrate that right now. We'll start with a rootstock liner, and this will be planted in the ground, and this is the ground line. And a very expert person will make a incision like this, and another incision like this. Hold on. And expose the cambium, okay, just like that. And then they'll go back and they'll have a bundle of bud sticks. And the buds have they're not quite completely developed, but I think I can find one that is somewhat ready over here. They'll take a bud, and this is what the next year's bud looks like. And they'll make the same incision on that bud and cut it off the tree, cut it off the branch, just like that. Okay, and voila, here is the bud, and now we're going to join it just like that with the rootstock. And this gets tied and covered for a few weeks, and when all of the tissues have developed it looks like a scab essentially. Basically, it's parenchymatic tissue that develops. Once it's all healed up, we overwinter the rootstock, and then cut the rootstock right here, and this new bud will become the new tree, and we'll have a new Fuji apple tree. Now, if you wanted a Fuji on one side and a gal on the other side, just put a Fuji apple on one side and a gal on the other side. I've seen people do that. But this is the way it's done commercially. And there are probably 25 to 30 million trees produced in the US every year. And the vast majority of those trees are produced by this simple method, by budding. It's hard to find out what's going on in the root, because when you pull out a root, it's like full of dirt and stuff. And so what we're doing here is we're actually growing apple trees, those small apple trees, in an aeroponic system that was developed, well, the concept was developed by NASA, but these systems were developed for the cannabis industry. And we're finding out all kinds of good stuff, from gene expression to metabolomics and hormones and all sorts of good things about the physiology and the genetics of apple roots. The very last point I would like to make is, why rootstocks? And why breed rootstocks? And it turns out that it is the most efficient way to breed fruit trees, and actually to breed other crops. Other crops are actually implementing rootstocks in their breeding scheme. Why do you think it's so important to breed rootstocks? Well, you're achieving an increase in efficiency because you're not selecting for all the root traits that you would be selecting for if you were breeding a whole plant. And so you're separating the root traits from the traits that make this apple very, very tasty and good. And also other traits that belong to the top part, the scion. And thereby gaining a huge amount of efficiency. So imagine breeding 10 traits in the scion: fruit shape, fruit color, let's see, what other things do they have? Bricks, so sweetness, the crunchiness of the apple, and all of those are controlled by many, many genes. So imagine selecting for all of those traits and then having to add fire blight resistance and then resistance to Phytophthora, resistance to Pythium, resistance to Rhizoctonia, and then the complex replant disease resistance, and then dwarfing. And dwarfing is two and a half genes. I know half a gene doesn't sound right, but there are slightly modifiers. And then you start breeding for the root system to absorb more potassium, be more efficient with calcium absorption, and all these other traits that are mounted up. And if you had to combine both scion and rootstocks together, it'd take more than 50 years. Or a couple million plants and some very smart guy doing whole genome selection. Instead, the two items are separated and then joined by grafting.
This has been a production of Cornell University, on the web at cornell.edu.